Okay, so we got a bunch of really interesting stuff sent in about this acetylcholinesterase, ACE. And
It's a lot of people are talking about it.
it's pretty amazing when you think about it, how our nerves, talk to each other, both like in our brains and throughout the rest of our bodies.
Right.
And this enzyme is like super important for making that whole thing work.
It's a big one.
Yeah.
It's, I mean, basically, it's kind of the traffic cop, right, for
Yeah.
your nervous system. I mean, everywhere you have two nerves meeting or a nerve meeting a muscle, you're going to find this ACE hanging out. And
Oh, wow.
it's basically breaking down acetylcholine, which is, that neurotransmitter. So it's stopping the signal like super fast. Like think of it as the off switch for your nerve impulses.
So it's like essential for things not getting all like jammed up.
And just firing constantly.
Wow, that's pretty wild. And so, not surprisingly, it seems like it's a big deal when it comes to medicine too.
Oh yeah, huge.
We saw that messing with AC can be really, really bad news, like with like nerve agents.
Right, like sarin and soman.
Yeah.
Those are, I mean, that's like the textbook example
Yeah.
of how powerful this enzyme is. I mean, if you block it, everything goes haywire. Like you have this crazy overstimulation of your nervous system. Like it's really scary stuff.
That's intense. But then, on the other side of that coin, it's also used in some pretty important medicines too, right?
Totally.
We read about phystigmin, neostigmin, and they're used for things like glaucoma, myasthenia gravis.
Right.
And then there's even research on this 1, 2, 3 hours for tetrahydronine aminoacridine, and that's for Alzheimer's. So like it's
because in that case you want to actually boost the acetylcholine levels.
Oh, interesting.
Right. So you kind of.
So it's like all about finding that sweet spot.
Exactly.
Yeah.
Exactly. Like how much.
Like Goldilocks and the Three Bears.
Yeah. How much do you inhibit? How much do you let it go? And it can be used for all these different things.
That's amazing. And it's pretty cool how I guess you could say they've used this knowledge to make insecticides like malathion that target ACU, but specifically in insects.
Yeah, and that's
so like how do they do that?
Well, it all comes down to subtle differences, right, between our enzymes and the insect enzymes.
Oh, okay.
And so they can kind of take advantage of those differences, make something that's hopefully safe for us. But really messes with the bugs.
Wow, that's pretty neat. But for as much as we were reading that there's still a lot that we don't know about how AT actually does its thing.
Oh yeah.
Like what are the actual nitty-gritty details of like the chemical steps?
Yeah, it's tricky because AC is so incredibly efficient, right? Like it works so ridiculously fast.
Yeah.
And so some of those like first initial steps of the reaction, they're like lightning fast, almost impossible to actually see.
Okay.
So it makes it hard to isolate, like the actual chemical breakdown.
Yeah.
It's like
so like those high-speed cameras they used to film, like hummingbirds or bullets.
Exactly. You need something even faster.
Yeah. So how do scientists even begin to tackle a problem like that?
Well, one really important concept that popped up a lot in the research you sent was this idea of a virtual transition state.
Virtual transition state. Okay.
Yeah, and it's kind of a mind bender, but think of it this way. So the enzyme's job, it's not just one thing.
Right.
Right. It's like a whole sequence of really, really tiny steps.
Okay.
Like a super fast, intricate dance.
Okay.
And each one of these steps, it has its own like high energy point.
Oh, yeah.
A transition state.
Yeah.
But when you have a bunch of these steps, and they're all kind of important for determining how fast the whole thing goes, what you actually observe, it's not like a single clear transition state, but it's kind of like an average of all these little tiny hurdles. And so that's what they call the virtual transition state.
So it's like we're seeing like a blurry picture because it's happening so fast.
Right.
And we can't slow it down enough.
To see each step?
It's like trying to watch a really complex dance move.
Yeah.
But you only see the beginning and the end.
Okay.
And you're missing all those subtle shifts in between.
Okay.
And actually this is kind of similar to some of the ideas that Northrop was working with.
Okay. Back in the day.
both of these approaches help us understand how different parts of the enzymatic process contribute to the overall rate.
Okay. And how we can use cool techniques like isotope effects to kind of dissect these different contributions.
That's fascinating. So then what does this virtual transition state idea actually tell us about HE itself?
Well, for one thing, it highlights that the active site, that place where all the action happens, it's not just like one spot. It's got different regions. You know, you've got your estratic site, your anionic site, and then you've got these hydrophobic areas.
Okay, and so those are all doing like different things to help this.
They all have different roles to play in the breakdown process.
Okay.
So the estratic site, that's like the business end, right?
Yeah.
That's where the bond actually gets broken.
Okay.
The anionic site, because it's negatively charged, it helps to attract and position these positively charged molecules.
Oh, okay.
Like acetylcholine. And then you've got these hydrophobic areas, you know, they're kind of water-fearing.
Okay.
And they help ACE to interact with other types of molecules that aren't charged. So it's really versatile because of all these different regions.
Wow. So it's not like a one-trick pony.
No, it's very multi-talented.
It's got a lot of tools in its tool belt.
Exactly.
That's pretty cool. And speaking of talents, it sounds like ACE is also like quite the contortionist. Like there's evidence that it actually changes shape.
Oh yeah. It's a very flexible enzyme.
Okay.
It's not just like a static, rigid thing.
Mm-hmm.
And that flexibility is actually really important for its function.
Okay.
You know.
So like what does that flexibility actually do like during the process?
Well, they think that it helps the enzyme kind of guide the substrate molecule through the reaction, you know.
Okay.
And it also helps to stabilize those fleeting transition states.
Yep.
We were talking about those virtual transition states.
Yeah.
So it's really key, and they've shown this. You know, they've done studies where they've used inhibitors to lock the enzyme in different shapes.
Yeah, wow.
And they've even made like engineered versions of the enzyme.
Hey, wow.
Where they've kind of, you know, tweaked its flexibility, and these studies all point to the same conclusion.
That it means
flexibility is important.
To be flexible.
Yeah.
Now we also read about how scientists are learning about this virtual transition state idea from studying how AC breaks down a specific type of compound,
right?
Called analytes, right?
Yes.
And what was it about those molecules that made them useful?
Well, what's interesting about analytes is that for those, it seems like both the initial binding of the molecule to the enzyme, you know, which might involve some of those shape changes.
Yeah.
And also the actual chemical step, the bond breaking step. They're both important for determining how fast the overall reaction goes.
Oh, so it's not like just one or the other.
It's a combination.
It's both.
And so the virtual transition state concept, it helps us kind of, you know, weigh the contribution of each of those steps.
Okay. So then how do scientists actually get down to like the nuts and bolts of these transition states? I know we read about this thing called pH rate profiles and solvent isotope effects.
Right.
What are those all about?
So those are really powerful tools.
Okay.
Basically by looking at how the rate of the reaction changes when you change the acidity, or when you swap out regular hydrogen for a heavier isotope like deuterium.
Okay.
You can actually get clues about which amino acid building blocks in the enzyme are actually involved in the catalytic process. You know, especially through the transfer of protons, which often involves, you know, residues like histidine.
Oh wow.
And you know, proton inventories. That's a really cool technique.
Okay.
Because that helps us actually count how many protons are being transferred during those critical steps.
So it's like a fingerprint almost of the reaction.
It gives you insights into the mechanism.
So to kind of bring it all together, like what have we learned about ACE today?
Well, it's a remarkable enzyme. You know, it's got this really complex active site with all these different binding regions.
All those little pockets.
Constantly shifting its shape.
Yeah.
And to understand how it works, we need these these really sophisticated concepts like this virtual transition state where multiple tiny steps are all important.
It's like a super highway, but with all these little tiny
little checkpoints along the way,
exits and things.
They're all influencing the traffic flow.
Wow. That's pretty mind-blowing.
And then, we've got these amazing techniques like these isotope studies that are really helping us to uncover these secrets.
It's amazing how much work goes into figuring out like one tiny little enzyme.
Yeah.
And I guess it makes you wonder like if HEE, which you might think is pretty simple,
it's
right?
like, break down this molecule.
Yeah, like how hard could it be? But then it's got all this incredible complexity.
And complexity too.
I mean, what other enzymes are out there that we might think are just kind of plugging along doing their jobs,
right?
But actually have these really incredible dynamic mechanisms that are just waiting to be fully understood.
I mean, that's what's so exciting, right? Like the more we dig into these biological systems, the more we find these layers of complexity that are just mind-boggling.
Yeah.
And who knows what we'll discover next.
And I guess, learning all of these tiny little details about how these enzymes work could really change the game when it comes to drug design too, right?
Oh, absolutely. I mean, right now, a lot of drug design is focused on targeting the active site.
Right.
That place where the molecule binds and the reaction happens.
Find that key, find that lock, and you're good to go.
But what if we could go beyond that?
Yeah.
What if we could take advantage of these more subtle interactions, these dynamic changes in shape,
have that flexibility.
these other factors that contribute to how the enzyme actually works. I mean, that could open up a whole new world of possibilities.
That's pretty amazing. Well, thank you so much for joining us for this deep dive into the world of ACE. It's been really eye-opening.
My pleasure.
And for everyone listening at home, we hope this has given you some food for thought. And until next time, keep on learning, keep on questioning.
Right.
And we'll see you soon.
See you later.